All right, thanks, Charlie. So today's talk is about the role of machine learning in targeted protein degradation. So as many of you are familiar with, the Ubiquitin-Proteasome system is a major pathway for selective protein degradation. And the specificity of the process is usually governed by E3 ligases that recognize either a sequence or structural motif in the substrates called the Degron, shown here in red. And due to the induced proximity with the E2 conjugating enzyme, ubiquitin can be covalently transferred, ultimately potentially resulting in polyubiquitination, which can then be a signal for proteasomal degradation. And like many biological processes, this has negative regulators as well. And in this case, they're deubiquitinating dubs that remove the ubiquitin, preventing, in this case, proteasomal degradation. And so given the importance of the UPS, it's perhaps not surprising that the UPS is frequently dysregulated in cancer, either in the UPS genes themselves, such as the well-known early cases of VHL and FBW7 being dysregulated, or in the substrates they recognize, such as beta-catenin or NRF2, which can escape protein degradation and thus have greater protein expression levels. We've seen in the past TPD docs numerous ways to either target or hijack the UPS system, and that could be through polymerization dependent degradation for a BCL6 degrader. It could be selective dub inhibitors and new molecular glue mechanisms as well. And we've even seen PROTACs starting to enter the clinic with AR and ER degraders and 11 more by the end of this year. But one of the more outstanding questions is, what will the future success of TPD be? And will it match the previous pace of FDA approvals for previous technologies such as antibodies? And I think the answer in part lies on us advancing the toolbox of targeted protein degradation, but it also depends on understanding what is the right target for the right patient. And since previous talks have extensively covered how to expand the TPD toolbox, my talk will be more about systematic approaches, especially computational ones, to find promising targets. So one way to try to find targets is to look for mutated genes that lead to disease, such as those reflecting the somatic evolution of cancer. And here I just show you the most prototypical progression of the normal colon epithelium all the way to colorectal cancer, which is typically associated with APC gene mutations, followed by, for example, KRAS mutations. And this was known way back in the late 1980s and early 1990s. But given the widespread DNA sequencing of human tumors, the breadth of how we understand mutational drivers has expanded considerably. For example, as a part of the Cancer Genome Atlas Consortium, we previously identified a consensus of approximately 300 cancer driver genes across 33 different cancer types. And to do this, we use 26 different computational methods, and there's a variety of ways you can tackle it. Some of the most common ways are to use a simple mutation rate model, where you're just looking at the number of mutations observed in tumors compared to some estimated background rate. However, there are limitations, and one of those is that they often have limited statistical power, as you can see here by the ever need for growing sample size to find more driver genes and more driver mutations. An alternative approach is to try to leverage more biological information in computational models. And one approach is to leverage the clustering of mutations in protein structure. And you can see for this example, the mutations shown in this spherical amino acid representations cluster at the interface between PIK3CA and PIK3R1. And this allows you to identify mutations that may be rarer, but are close in space to more commonly mutated driver mutations, and thus may implicate them as also likely mutational drivers. And if you take these types of approaches, you see that the validation rate in our previous TCGA PanCan Atlas study was approximately 75% in cell viability assays, which was greater than many alternative approaches. And there's been a variety of methods that have been developed, including our own, as well as those developed by the Li Ding lab, like Hotspot 3D. But what's really fundamentally missing from these models is really anything capturing the human disease biology of protein degradation. And if we knew every oncogene where protein stability was a critical dependency, then it would be considerably easier to know when PROTACs or other degrader drugs may have unique advantages over perhaps other inhibitors or other induced proximity-based drugs. So we can try to hone in on the disease-causing proteins where protein degradation might be more promising. So in this talk, I'll cover how can we use machine learning in conjunction with experiments to uncover the disease biology of protein degradation, and particularly for cancer. This will be in two parts. In the first part, I will cover the leading role of machine learning in going from mutation to mechanism in terms of understanding human tumors. And so towards this end, we developed a variety of computational methods to dissect how mutations may shape the cancer proteome by altering protein degradation. And this involves understanding each component of the UPS. That is, the genes within the pathway, their substrates, which you can see here, and the relationship between the two, linking the UPS genes to their substrates. And I'll start off with trying to define the set of UPS genes that may be implicated in driving cancer growth. So to do this, we examined which UPS genes were somatically mutated more often than expected across the Cancer Genome Atlas. And you can just visually see the pattern of mutations from this lollipop diagram, which shows individual mutations with these circle markers along the protein sequence, of this case, just one example of FBW7. We use a variety of features to describe the pattern of mutations, including the clustering of the mutations, how close are they together, the types of consequences, the evolutionary conservation, and many additional covariates. And then we relate this knowledge to knowledge in the literature of oncogenes and tumor suppressor genes, which allows us to train a machine learning algorithm we call 2020+ to make predictions on unknown genes on whether they're an oncogene or a tumor suppressor. So here I've plotted the results from 2020+ across TCGA. And you can see on the x-axis, you have the maximum oncogene score across the various cancer types. And then on the y-axis, you have the maximum tumor suppressor gene score from our predictions. We identified 63 genes as statistically significant, marked in red. And of those, 33 were not previously reported in other studies. And this was partly due to us focusing on UPS genes and therefore increasing our statistical power. The majority are E3 ligases at 73%, and also a substantial percentage of deubiquitinating enzymes and a much smaller percentage of E2 or E1 enzymes. And if you were to tally this up, it represents approximately 16% of all cancer driver genes in the TCGA, which is quite substantial and comparable to many other prominent pathways. However, one of the kind of issues that you can see here is that most of these predicted driver genes are actually predicted to be tumor suppressors. And you can see that from many of the significant hits here having a high tumor suppressor gene score compared to an oncogene score. And so they would be less attractive per se as targets because you'd have to restore their function, and perhaps targeting their upregulated protein substrates might be a better way to approach this. And so that's why next we wanted to identify what are the substrates underlying these UPS genes that we found as likely cancer driver genes. So one approach to do this would be to look at protein expression changes when a UPS gene is mutated and whether a putative substrate went up or down. However, there's really not that many proteomics data sets available in cancer. So we had to come up with an alternative strategy. And we reasoned that transcription factors might be particularly useful to study. That's because the RNA expression of a transcription factor target genes could really serve as a proxy for the protein abundance of a transcription factor. And then we also adjust for a variety of covariates, including the RNA expression of the transcription factor, to try to avoid artifactual associations. So we do this by building differential expression signatures of the mutant compared to the wild type that adjust for the aforementioned covariates. And then we want to infer what are the transcription factors that may explain the differential expression profile, either ubiquitin ligase that may be negative regulating a transcription factor, or perhaps a D-ubiquitinase that may be essentially a positive regulator of the transcription factor. So to do this, we leveraged all of the ChIP-seq data from the full systrome database containing thousands of ChIP-seq profiles. And what this does is it catalogs the DNA binding of transcription factors. And the hypothesis is that if a transcription factor binds near a gene in the genome, then it's more likely to regulate its transcription. And we can combine this information with the differential expression profile using the method Rabbit developed in the Lu lab. So Rabbit found many significant hits that were associated with the mutation of UPS genes, which you can see here. Well, on the y-axis is the minus log 10 transformed p-values, and the x-axis here is the various UPS genes, and each dot represent a UPS substrate putative pair. And we decided to focus on the top 100 non-redundant associations from our analysis, which you can also see on the right. So our analysis found many direct hits and nearby indirect hits that are known in the literature, such as FBW7 and MYC, SPOP and androgen receptor, CYLD and NF-kappa B signaling, and VHL and HIP1 alpha, although we found the binding partner to HIP1 alpha aren't. So as an example, our analysis confirms a previously known relationship of KEAP1 regulating NRF2 encoded by the NFE2L2 gene. And since COL3 is a necessary scaffold for KEAP1, people have really previously assumed that COL3 mutations only really are oncogenic through the NRF2 protein level as well. But through our analysis, we found that not only NRF2, but also MYC was a significant hit in our results. And so this suggested to us that there's probably likely a second target recognition subunit here that could recognize MYC. So Xiao Qing, a joint postdoc in the Shirley Liu lab and Miles Brown lab, decided to investigate whether this relationship was actually a direct mechanistic one. And by knocking out COL3 in head and neck cancer cell lines, which is where this association was found, he found that MYC protein half-life increased, as you can see here, by following a translation inhibition by cyclohexamide. The MYC protein level remained fairly stable as compared to the control condition, which is MYC is rapidly degraded. And furthermore, C-Myc co-immunoprecipitated with COL3, suggesting that this is likely a direct rather than an indirect mechanism. So we really think that our statistical analysis of transcription factors is enriched for direct mechanistic relationships and may be worth further studying. So next, I'll talk about how can we use new computational methods to help us unbiasedly understand the sequence code of Degrons that control protein degradation. So this will be all about the substrates and what mediates the recognition for degradation. So to do this, we collaborated with the Steve Elledge lab and his former postdoc, Richard Timms, now a group leader at the University of Cambridge. And they built an assay called GPS, short for Global Protein Stability, to measure the stability of all C-terminal and N-terminal sequences in the human proteome. And basically how this works is you have known sequences fused to GFP, which fluoresces green, and you compare this to an internal control protein, DS-red, that fluoresces red. And Richard could then measure stability through sorting cells by FACS based on the red to green ratio, and then quantify the relative stability through sequencing. So this systematic assay motivated us to develop a neural network model called Deep Degron to predict the underlying sequence motifs known as Degrons that target proteins for degradation. And from this diagram, you can see that the input is protein sequence, and then the output is whether the sequence is likely to be highly degraded or not, according to the GPS assay. And we chose to model this a two-layer fully connected neural network with ReLU activation units. A second thing to note is that because some of this prediction may come from generally stabilizing amino acids, regardless of whether there's an actual Degron motif, we also trained a second model that didn't have any of the sequence order information and subtracted that out from our full model. So subtract out the background protein stability effects of amino acids to hopefully leave what we call a Degron potential that reflects sequence-specific motifs that may be in the peptide sequence leading to its degradation. So before we wanted to try to predict whether mutations may lead to the loss or gain of Degrons, we wanted to see whether Deep Degron was highly accurate or not. And so the first thing we did was evaluate Deep Degron on a held out test set. And you can see the receiving operating characteristics curve with the sensitivity here on the y-axis and 1 minus the specificity on the x-axis. And the Deep Degron had high performance according to the area under this curve of 0.93 out of 1.0, indicated by this blue curve here. And then notably, it was considerably better than the previous rule-based alternatives proposed in the literature, such as just considering top 100 motifs shown in purple, as well as also considering just the composition of amino acids, like the number of bulky amino acids and number of acidic amino acids, shown in these two curves here, and then better than the combination of these three rule-based approaches with logistic regression shown in this orange curve. So to validate whether Deep Degron could really identify new Degron motifs, we sought to test 21 novel Degron motifs and whether mutation of these motifs would lead to protein stabilization according to the flow cytometry results. What we found was that 67% of these putatively novel C-terminal Degron motifs validated, and 89% of the N-terminal motifs validated. And you can see that here by the shifting of this kind of salmon-colored curve to the right compared to the wild type construct in gray. So given that we were now reasonably confident in the performance of deep Degron, we wanted to examine for genes that may have had an enrichment for Degron loss mutations in human tumors in the TCGA. And so when we analyzed this, what we found was that GATA3 and PPM1D showed the largest evidence for Degron loss. And you can see this from a large negative change in the Degron potential that I mentioned earlier, as well as it being statistically significant according to our analysis. So we decided to experimentally validate both of these predictions. So PPM1D is amplified or mutated in a variety of cancer types, including breast cancers, pediatric brain cancers, and myeloid neoplasms. And PPM1D is thought to be an oncogene, as it's a phosphatase known to be a negative regulator of p53. In the case of myeloid malignancies, the Goodell and Ebert labs associated with prior chemotherapy treatment, as you can see with enrichment for PPM1D mutations and therapy-related myeloid neoplasms from this plot. And their evidence suggested that PPM1D mutant proteins had elevated protein stability, allowing cells to survive cisplatin chemotherapy treatment. Although really the exact mechanism behind how this happens wasn't really clear. So most of the PPM1D mutations are truncating mutations near the C-terminus, including frame shift and nonsense mutations that would eliminate the C-terminus of PPM1D. And according to our predictions, this would eliminate a C-terminal Degron that contains a VC motif. So Xiaoqing therefore tested whether point mutations of these putative Degron would lead to increased protein stability. And what he found was that really, as expected for a Degron loss event, either point mutation led to increases in protein expression of PPM1D to a similar level of truncating mutations that are similar to what are observed in human tumors. And then furthermore, as expected for PPM1D's role in the DNA damage response pathway, we see there's greater dephosphorylation of ATM and P53. So this suggests that loss of a C-terminal Degron, at least in part, may contribute to phenotypes that have been reported in the clinic. We next examined GATA3, which actually showed the largest evidence for Degron loss. And GATA3 is mutated in approximately 13% of breast cancers, making it the third most frequently mutated driver gene in breast cancers, and is typically associated pretty much almost exclusively with the estrogen receptor positive subtype of breast cancer. As you can see, in this case, GATA3 has a substantial amount of frame shift indels, shown here in red, at the C-terminus. However, people have thought that actually maybe GATA3 is a tumor suppressor gene, because these frame shift indels normally people would assume are loss of function, and therefore GATA3 should be a tumor suppressor. But if you remember from the PPM1D cases, this distribution of mutations is actually really strikingly similar. In that case, it's more relatively known that PPM1D would be more of an oncogene role. Actually, consistent with our prediction of Degron loss, if you look at protein expression level in human tumors from reverse phase protein arrays, you see that GATA3 frame shift mutant tumors, shown here in orange, have higher protein expression levels than their wild-type counterparts. And this is even true after you adjust for tumor subtype and RNA expression of GATA3. And interestingly, GATA3 had one of the most enriched Degron motifs, according to our analysis, which consisted of a glycine at the very C-terminus and an alanine at the third to last position. So to examine this further, Xiaoqing wanted to mutate each of these residues to see what the effect on protein expression was. And what he found was that mutating either of these two residues that we identified from deep Degron led to increased protein expression levels, similar to a truncation of GATA3. And furthermore, if you were to inhibit with the proteasome, you see that all isoforms lead to upregulation, consistent with this being a protein degradation mediated mechanism and likely a Degron that's being lost by these frameshift indels. We next look for what the potential functional effect of Degron loss on GATA3 could be by examining ChIP-seq data. And what we found was that mutating the Degron on GATA3 led to elevated DNA binding. And these upregulated DNA binding sites from ChIP-seq were preferentially near estrogen signaling genes. We also confirmed that this led to upregulation at the protein level to Western blots as well. And so this is consistent with where these GATA3 mutations are found in human tumors, and that's in the ER-positive, estrogen receptor-positive breast cancers. So really, if anything, these experiments are consistent with both a function and a phenotype that are reflective of GATA3 as an oncogene. So if we now take this perspective that GATA3 is an oncogene because of elevated protein expression, the variant interpretation changes considerably. First, these C-terminal frameshift mutations are likely gain of function because they would eliminate a C-terminal Degron at the very end of GATA3. And this also likely explains why there's no frameshift mutations at the end terminus of GATA3, because this would eliminate the entire protein sequence of GATA3, be loss of function, which is a passenger event in an oncogene. So we next wondered, do genetic fusions like point mutations also frequently result in the loss of Degron? And so gene fusions are often the result of chromosomal translocations, where you can see in this example, breaks in chromosome 9 and chromosome 22 can lead to a rearrangement that's known as the Philadelphia chromosome, ultimately resulting in the BCR-ABL gene fusion composed of a sequence of two distinct genes, BCR and ABL1. And much of the theory around the oncogenicity of gene fusions encompasses several mechanisms, including increased transcription via promoter swapping or enhancer hijacking, as well as increased dimerization of proteins leading to greater protein activity. And so we reasoned that Degron loss could be another widespread mechanism. However, there are only a couple reported instances of Degron loss in fusion events in the literature, such as the loss of top 1 Degrons in ETV fusions, reported by the Vishva Dixit lab, and the loss of S-pop Degrons in a subset of ERG fusions reported by the Wenyi Wei lab, both in prostate cancer, unclear whether it would be a broader phenomenon. And you can see this in this diagram here, that originally ETV1 would have 2 Degrons here, and then the fusion with TMPRSS2 would lead to the loss of these Cap1 Degrons. And because of the sparsity of these Degron loss events reported for gene fusions and standing really in stark contrast with other types of mutations in cancer, we thought this might be really an artifact of a lack of a systematic analysis of Degrons, rather than reflecting anything biologically meaningful. So towards this end of doing a systematic analysis, we trained a machine learning model called random forest to predict Degrons in protein sequence. And to do this, we used 86 features describing the evolutionary conservation at that location within the protein, various predicted biophysical features, including solvent exposure, flexibility, secondary structure, et cetera. And using this information and cross-validation, our model had a good performance with an area under the receiving operating characteristics curve of 0.81 out of a maximum of 1.0. So with this, we wanted to now systematically analyze the more than 20,000 gene fusion events in 9,000 tumors found in TCGA. And based on the approximately 250 Degron motifs that either we learned from the C-terminal Degron analysis or were known in the literature for E2 ligases recognizing internal Degrons, we predicted the likelihood of each motif match across the proteome as serving as a Degron using our machine learning model to try to really prioritize the ones that could actually be recognized, that may be on the surface in the right position, such that an E2 ligase could recognize a motif if it saw it. And so with this, we identified 2,400 fusion events that were predicted to lead to Degron loss. And interestingly, there was a clear and significant bias for Degron loss in fusions containing oncogenes compared to tumor suppressor genes, where approximately 30% of recurrent oncogene fusions were predicted to lead to Degron loss, and you can see this in blue, while the opposite, that Degron gain, tended to be more true for tumor suppressors. So we next want to identify what are the specific genes and fusion events that preferentially underwent Degron loss, instead of just examining oncogenes and tumor suppressors in aggregate or in general. And so using the set of predicted Degrons, we calculated a Degron loss score for each gene. And this reflects both the confidence in our machine learning prediction. So for example, here, the machine learning algorithm may have given a score of 0.7 out of 1.0 for this putative Degron, but as well as the frequency by which is lost in fusion events. So if you have multiple fusion events repeatedly leading to Degron loss, say in this case you have 3, then the Degron loss score would be 3 times 0.7 or 2.1. And then by using a permutation test, we could then evaluate whether the Degron loss score was greater than expected by chance using a false discovery rate of 10%. So from this, we identified 58 genes with significant enrichment for predicted Degron loss in TCGA. And here I plotted the most recurrent fusion events for each cancer type and labeled fusion events with a color dot if they were identified as being enriched for Degron loss according to our statistical test. And encouragingly, we could recapitulate the enrichment for Degron loss for ETV fusions of top 1 Degron. But surprisingly, many of the most recurrent fusion events were predicted to have Degron loss, but weren't reported in the literature as such, including EGFR fusions, PML-RARA, BCR-ABL, RET fusions, and RAF1 fusions. And while we experimentally validated the effect of Degron loss for these 5 fusion events that I've highlighted, I'll just cover one example, BCR-ABL. So for the BCR-ABL fusion, you can see that the first 928 amino acids of the BCR-ABL, or the BCR protein is maintained. And you can see this by this black line going to the 928th position, while only the first 27 amino acids of ABL1 protein is lost, indicated by the fusion junction right here. And really, despite this, a high likelihood SPOP Degron, according to our machine learning predictions, occurred just proximal to the fusion junction, resulting in its putative loss in the ultimate fusion gene product, which you can see is absent from the BCR-ABL gene fusion. Consistent with this prediction, Jing Liu from the Wenyi Wei lab found that the stability of wild-type ABL1 is increased when SPOP is knocked out, and you can see that in the orange curve here after cyclohexamide treatment, it's relatively stable protein expression compared to wild-type SPOP in blue going down. Furthermore, deletion of the predicted SPOP Degron led to stabilization of the ABL1, which was in stark contrast to the wild-type ABL1. And then lastly, but most importantly, the BCR-ABL fusion is resistant to SPOP-mediated degradation, which you can see here that contrasts with the original wild-type ABL1 protein. And combine this with additional data that it's not shown here, that indicates this is COL3 and proteasome dependent. This suggests BCR-ABL fusions lead to the loss of the sole SPOP Degron in ABL1, and consequently to increase protein stability for this oncogenic gene fusion. So to summarize the first part, new computational methods can be really essential for discovering new human cancer biology of protein degradation. And protein degradation constitutes approximately 19% of all driver genes in cancer, a fairly large percentage. And machine learning can really help try to take more unbiased approaches to reveal mutated Degrons implicated in cancer, indicating disease-causing proteins that have higher stability and perhaps would be attractive for targeted protein degradation-based approaches. So in the second part, I'll cover how machine learning can help understand results from phenotypic screens, such as those for immunotherapy response. So this project was led by Xiaoqing Wang, and what he did was conduct an in vivo CRISPR knockout screen using a syngeneic triple-negative breast cancer model across 4,000 genes in his initial screen, and then subsequently a secondary screen at a much more targeted focus set from that 4,000. And to understand whether the tumor growth was impacted by the immune system, he compared new T-cell deficient mice to mice with varying levels of immune competency, including wild-type mice, as well as anti-PD-1 treated mice. And what Xiaoqing found was that guide RNAs targeting the E3 ligase COP1 were the most significantly depleted hit in the screen, but this was really only true for immune competent hosts, suggesting that perhaps COP1 was mediated through the immune-mediated mechanism. Consistently, individual knockout of COP1 rather than a pooled screen setting led to reduction in tumor growth, as you can see by the orange and red curves being lower, both in the wild type and anti-PD-1 treated mice, which is not true for the new T-cell deficient mice. And by immunohistochemistry, he found a significant depletion of macrophages, which was also consistent with the flow cytometry results. And you can see that here for the control, there's substantial staining in brown for macrophages, but for COP1 knockout, there's considerably less. RNA-seq of COP1 knockout cells indicated that macrophage chemoattractants were among the most downregulated genes, indicating that although COP1 knockout occurred in the cancer cells, it likely could influence the tumor microenvironment through secretion of chemoattractants. And this downregulation of macrophage cytokines was further supported at the protein level from protein array data. So given this unexpected immune-mediated phenotype of COP1 knockout, we were naturally curious what COP1 substrate or substrate underlies this phenotype. And there are numerous reported substrates in the literature for COP1, which has led to considerable debate on whether COP1 is an oncogene or a tumor suppressor based on the particular substrate. Much attention has been given to the opposing roles of P53 and Jun, although probably the more overarching trend here is that many of these substrates are transcription factors, such as the transcription factors S1 and S2 found by the Collin lab, or CBP alpha found to be important in AML. And there are generally two mechanisms by which COP1 acts. One is a Collin-dependent or Collin-based mechanism, where COP1 serves as an adapter for the Col4-Det1 E3 ligase to recognize substrates like ETV1 and Jun. Another is a Collin-independent mechanism, which often needs additional adapter proteins in the triple pseudokinase family to recognize and degrade substrates often in the CBP family of transcription factors. And because our phenotype, particularly in cancer cells, really hasn't been previously reported, we suspected that our hit may be a result of a potentially new substrate that's not listed here. So to examine this, we again used our machine learning model to predict COP1 Degrons. And what we found was that only 7 proteins had a predicted COP1 Degron and were significantly upregulated in the COP1 knockout cells in the proteomics data. And you can see these 7 marked by a black outline of the dot in the scatter plot. And interestingly, TRIB2 was the most upregulated among those that had a COP1 Degron, which suggests it's likely serving as an adapter for the COP1 protein, and that perhaps TRIB2 might help mediate the protein degradation of a transcription factor similar to the previous substrates. To assess this possibility, we analyzed proteomics data with and without proteasome inhibition by MG132. And what we're looking for here is proteins, particularly transcription factors, that go up in protein expression, the control condition, indicated by having dark red. But once you knock out the E3 ligase responsible for its degradation, there's no longer upregulation when you inhibit the proteasome. And the only one that really fits this profile was CBP delta, which suggests that CBP delta might be degraded by COP1. Xiaoqing tested this hypothesis and found that COP1 co-immunoprecipitated with both TRIB2 and CBP delta, indicating a direct interaction. Knockout of COP1, as seen in these 2 lanes, led to increased CBP delta protein expression, and conversely, overexpression of COP1 led to a reduction of CBP delta protein expression level. And notably, this was not explainable by RNA expression of CBP delta and was proteasome dependent, indicating that COP1 likely degrades CBP delta. And since TRIB2 knockout also led to the abrogation of the interaction between CBP delta and COP1, indicated by the absence of a band here, as well as TRIB2 knockout led to an increase in CBP delta protein expression levels, it really suggests that TRIB2 is serving as an adapter protein for CBP delta protein degradation, mediating the COP1 mechanism. Phenotypically, in anti-PD-1 treated mice, we see that individual knockout of COP1 leads to inhibition of tumor growth, shown by this black curve. But if you double knock out CBP delta and COP1, it reverts it back to wild type growth, indicating that CBP delta likely explains most, if not all, of the phenotypic impact of COP1 knockout in this triple negative breast cancer model. And consistent with this, RNA-seq of CBP delta knockout cells showed the differentially expressed cytokines, and Kaplan knockout is really an opposite direction for CBP delta knockout, indicating it kind of is meeting the opposite effect, as you might expect. So to summarize the second part, we saw that COP1 degrades CBP delta through the adapter protein TRP2, which leads to altered macrophage infiltration and altered anti-PD-1 response. And we saw that machine learning can help prioritize Degrons that ultimately help identify the substrates underlying UPS gene hits in phenotypic screens. And then more broadly, it emphasizes that the in vivo and in vitro phenotypes of UPS genes can differ quite dramatically, just because of the impact of a single substrate. In this case, is CBP delta, and if you don't have mice with an intact immune system, you're not going to observe the same phenotype as ones with it. So to conclude, through both parts of the talk, we saw that machine learning can inform target discovery directly from human tumor profiles, as well as from phenotypic screens. And that trying to model each aspect of the ubiquitin-proteasome system can reveal unexpected findings, such as thought to be tumor suppressors that might actually be oncogenes because of the unappreciated role of Degron loss for example, for frame shift indels. And I think we're heading towards an integrated computational understanding of the human biology of the ubiquitin-proteasome system and disease, as well as with targeted protein degradation-based drugs, where ultimately the intersection between the two might yield the most promising benefit. And so lastly, I just want to acknowledge Shirley Liu and everybody else in the Liu lab, including Wu Bing, Xiao Qing, who's jointly in the Liu lab and the Miles Brown lab, who did much of the experimental validations, the Wenyi Wei lab, and Jing Liu, who examined the gene fusion events, as well as the Steve Elledge lab and Richard Timms, who looked at the GPS and C-terminal Degron analysis, and lastly, the Eric Fisher lab, and my funding from the BCRF Foundation and the Damon Running Cancer Research Foundation. And thank you for listening.